Thank you, Adam and Gord. It's a great pleasure and honor to be here talking to you about the pet subject of docosahexaenoic acid. And it really is a pleasure to be back with a lot of friends from Gord and the omega-3 research establishment. And I want to really centralize on docosahexaenoic acid because, now how's this? It's not working. Hello? Can we get something to happen? There we are. Anyway, the point I want to make is that the brain evolved in the sea some 500 million years ago using docosahexaenoic acid, and it's never used anything else since then, despite all the genetic change that has taken place over that 600 to 500 million years. So there's something very important about the use of docosahexaenoic acid in the brain, particularly as it's the brain that makes us different from other animals. And something that people really have forgotten is that Charles Darwin, in his all 6 editions of the book, said there were 2 forces in evolution, natural selection and the conditions of existence. And he said, again in all 6 editions, that the most powerful of these was the conditions of existence. And so, to use John F. Kennedy's expression, we celebrate the past, that's evolution, to awaken the future. And DHA is and was a conditioning existence, a condition of hoar existence of Homo sapiens. And so that's really what we're going to be talking about. The condition of existence, I just like to use that picture. It's the beginning of stars and life and everything. Okay, now, the point really is that people have sort of ignored the significance of lipids in biology, thinking of them rather as bits of oil that separate chunks, cells from the outside. And we know, I'm not going to go into any detail about this because others will talk about it, but I'm sure you all know by now the metabolism of these essential fatty acids, the 2 families, linoleic acid and alpha-linolenic acid. And the point I want to make about this picture is that to develop docosahexaenoic acid, you not only have a very large number of limiting steps, which makes it very difficult to synthesize in large animals like ourselves, and therefore you need the preformed fatty acid in its own. But not only that, but I'll point out that it takes 6 oxygen atoms just simply to remove the hydrogens to form the 6 double bonds in docosahexaenoic acid, which will become quite important in a minute. Now, during evolution, life started about 3 billion years ago. But for the first 2.5 billion years, nothing much happened. It was basically photosynthesis, and the photosynthetic systems, of course, produced oxygen. And a whole lot of other things produced oxygen as well. But when you came to the Cambrian explosion, which was about 600 million years ago, you had a complete new life forms began. For the first 2.5 billion years, you can find no record in the fossil history of any intracellular detail. And suddenly, at 600 million years ago, you find intracellular detail. And not only that, we have the explosion of all 32 phyla that come into existence more or less instantaneously with the advent of oxygen. Now, everybody has talked about this as being an event created by oxygen and oxidative metabolism, which is much more efficient, 8 times more efficient than anaerobic metabolism. But I want to put another story to you, and it's this, that in fact, what's happened with this? Why doesn't this produce the whole thing? No. Can you go back to that last one? It's not reproducing the slide. Oh, well, never mind. In that back gap there, if the Macintosh could have reproduced what the Microsoft reproduces, you would have seen mitochondria. And mitochondria are, of course, full of structures. And round about the mitochondria, you can see the reticuloendothelium, and round about that lot, you can see the nuclear envelope, all neatly tied up, and the plasma membrane as well. And all these membranes, upon which the intracellular functions of the cell depends, are made with lipids. And people forget about this. But it's absolutely fundamental to this new form of life that occurred 600 million years ago, was the sophistication of organization within the cell, if you like, speciation inside the cell, which led to differentiation and speciation of the extracellular system, and hence the speciation in all 32 phyla that we have today. So the lipids played a fundamental role in allowing this higher organization that took place when oxygen became available. And I'll remind you that for docosahexaenoic acid, it requires 6 oxygens just to remove the hydrogens to make 6 double bonds. So the synthesis of lipids became supremely possible in the most complex manner, allowing for the complexity of structures that make up the cell membranes. Well, I'm sorry about the missing bit, but it all happened in the seas in the Panathalssic oceans 600 million years ago. And there's the eye of Orthoceras to show you that the eye, the visual system, really was one of the first things that happened, and it was almost pretty well complete in its evolution by 450 to 500 million years ago. So now let's just look at the photoreceptor. Now to just think about the extraordinary thing about the photoreceptor, what's happening, can you go back to the one before, please? The photoreceptor, the extraordinary thing is that it actually faces the wrong way. You would expect the photoreceptor to face the incoming light. Well, it doesn't. It faces the retinal epithelium. And that is strange, and nobody has an answer to that, but we'll come up with an answer to it as we go through the story. Now, the chemistry of the photoreceptor is absolutely dominated by docosahexaenoic acid. And the interesting thing is, if you look at cod mussel, you'll find that cod mussel in the green line actually is extremely rich in docosahexaenoic acid. The phospholipids of cod mussel, the structural things that are in the cell membranes, has about as much docosahexaenoic acid as you find in the photoreceptor. But it's in the photoreceptor itself, practically every molecule not only has docosahexaenoic acid in the structural cell membranes, but almost 2 molecules. Right. Now, I want to talk about what this might have meant through evolution, in the sense that it's docosahexaenoic acid and these lipids are not just playing a role in structures and in providing small molecules to do all sorts of exciting things. They're also intimately involved in influencing the genetic information. And we know that docosahexaenoic acid, for example, is a ligand for the retinoid X receptor, a pivotal nuclear receptor that influences gene function. And a colleague of mine, Andrew Sinclair, who worked with me for a number of years in Kajaka, in Szeged in Hungary, have looked at the way in which omega-3 fatty acids in particular, it's DHA when it gets to the brain, there's no other omega-3 fatty acids in the brain of any sort, actually influences, triggers the stimulation of, in the first paper that they published, some 107 different genes involved with brain growth and development. And that's the number of genes that they have reported. And in the detail, there's some of these are some of the genes that are switched on by docosahexaenoic acid by a factor of more than 4-fold. So in other words, what docosahexaenoic acid is doing through the nuclear receptors is actually influencing the way the gene behaves. And that's really an important issue because it explains the way in which docosahexaenoic acid was essential, ultimately, for cerebral evolution. And we'll come to that in a little while. Now, Andrew, again, has been doing some other interesting stuff with the histones. And what he has found just recently is that the histones, which are core features, protein features that surround the DNA and enable it to function, are actually influenced to a very powerful degree by zinc and docosahexaenoic acid. But the interesting thing that he has discovered is that if you look at this doesn't work properly, but anyway, on this side here, you'll see that docosahexaenoic acid plus zinc is operational, but zinc on its own is not. So this again talks about the way in which the trace elements are absolutely functional in participating with these essential fatty acids. And one of the things that's important about this is, of course, the sources of these trace elements, the richest sources of these trace elements, are the same as the sources of docosahexaenoic acid, namely the marine food web. And in summary, his work really shows that docosahexaenoic acid alone will influence the histone function. And of course, gene regulation goes up, and neurodevelopmental disorder goes down. So this is really quite an important advancement in our understanding of the way in which docosahexaenoic acid is working, not only on its own, but in cohort with trace elements. So, in other words, his conclusion is that docosahexaenoic acid may contribute with zinc to future treatment and prevention of neurodegenerative diseases. Well, that's for future research. Work that we've been doing, interestingly enough, has been to use electrospray mass spectrometry to look at the molecular species, because most people just simply, and we mostly in the past, simply looked at the fatty acids in omega-3 deficiency. And here you can see the impact of omega-3 deficiency in the mother on pups. The deficient pups have got that green amount, the blue amount of DHA, whereas the pups coming from mothers who have been fed with docosahexaenoic acid have very significant amounts. This is nothing new by any means, but with electrospray, you can actually look at the individual molecules, lipid molecules that are in these cell membranes. And it's just to sort of give you an example about this, what you can see here is that they're behaving in very odd ways, that some of them are behaving as you might expect. The linoleic acid derivatives, arachidonic acid, and so on, molecular species go up. But the stearic acid and arachidonic acid, there is absolutely no difference in time between the control animal and the deficient animal. And this is really fundamental, because we don't see this when you just simply do fatty acid analysis. What is happening is that the individual molecular species that are in these cell membranes have individual functions in themselves. And again, if we look at the docosahexaenoic acid system, you can see again you would expect docosahexaenoic acid going up in the open circles and in the deficient, but again you see extraordinary how the 18-1-22-6 is behaving quite differently to the 18-0-22-6 with or without for the feeding of docosahexaenoic acid itself. So the point I'm trying to make is that you cannot look at these lipids as just simply a bunch of fatty acids. They are specific molecular species that are having specific functions in relation to proteins and signaling systems in the brain. And if you look at what's been going on throughout the evolutionary history, if you take the eye spot of the dinoflagellate, for example, which lived 600 million years ago, you will find not only docosahexaenoic acid in the eye spot, but you also find didocosahexaenoic acid, as you've got in your own eyeball. And if you take go right through down from the dinoflagellates to the cephalopods, the fish, the amphibia, the reptiles, the birds, the mammals, it's always the same. And this, to me, is this a 600 million year period of evolutionary change. And there has been absolutely no change in the use of docosahexaenoic acid in the primary signaling system, which was the pivotal in the evolution of the animal systems that we came down to us. What effectively happened was that in the first period of evolution, the photosynthesis was producing carbohydrates and proteins, as green leaves do today, and excreting oxygen. And what happened at the Cambrian explosion was you had a completely different chemistry in which the reception of photons no longer turned the stuff into carbohydrates and proteins, but the energy produced electricity. And that electricity stimulated this evolution of the nervous system and ultimately the brain. And the chemistry of that system is the same today as it was 600 million years ago. So so far as I'm concerned, that really is the most compelling evidence for the pivotal role and primary requirements of docosahexaenoic acid for the developing nervous system, its growth, function, and maintenance. Now, if we look at the contemporary situation and look at In this case, Andrew and I looked at 42 different species that live on the planet. And we looked at the variability of their diets and the variability of their genes. And we looked at the variability in the docosahexaenoic acid, linoleic acid. You can see in this slide in the liver a huge variability amongst the animal kingdom. But when you look at the brains of mammals, that variability vanishes. So this again demonstrates the absolute pivotal requirement of these essential fats for the development of the brain and the nervous system. And that is what made us what we are. So what I really want to say to you is that this is a pretty significant feature in terms of the whole sort of understanding of genetic makeup and Darwin's conditions of existence as being the most powerful determinant of evolution. And when you test this in experimental animals, and people have done this with children, et cetera, what you see if you look at neurogenesis in the pup brain of a mother that's been fed docosahexaenoic acid, you see beautiful migration of the cells to form the cortex, whereas by contrast, where the mother is deficient, you see a very poor migration. And that can be explained through what I've been talking about the way in which docosahexaenoic acid is influencing gene expression in the brain, and it explains all the experimental studies done by Hiroma in Nagoya in Japan, by Eileen Birch, Bob Gibson, Forsyth, and all these other people that have looked at this in children fed on breast milk with or without, or breast milk formula with or without DHA. So you can see that that is the fundamental explanation for what is going on in terms of the way in which docosahexaenoic acid can influence cognitive function and in particular visual acuity. Eileen Birch says that if you feed a child or infant on a milk formula without DHA, the difference in visual acuity is 1.5 lines in the eye chart. It's as significant as that. And so really, in effect, when you think about it, throughout 600 million years of evolution, there's been no change, and there's been this dominance of docosahexaenoic acid influencing gene expression. And in fact, the conclusion you are forced to come to is that DHA was the master of DNA, very consistent with Darwin's first hypothesis that the conditions of existence is the most powerful factor in evolution. Right, let's just look at a few more bits about this. Peroxidation is a very interesting thing because Alzheimer's and special evidence for individual positions, there's special evidence coming up for not only this specificity of a molecular species I was talking about, but also the evidence for individual positions in the molecule as being critically determinant. Now let's just look at everybody knows about this, the arachidonic acid cascade, and so I'm not going to go into detail about it other than to say that the position there which you see as the bisalycyl position of the CH2 is where you get the first abstraction of hydrogen leading to the peroxidation of the molecule. And again, this is the same thing that you can actually get without enzymes, the synthesis of PGJ2 from arachidonic acid through that oxidative process. Now, we're not going to really worry about this, but again, we have another example with Nicholas Bazan, who's going to talk later, in the synthesis of neuroprotectin D1 from docosahexaenoic acid. So this position in the molecule is absolutely pivotal, and this is where the first attack occurs with peroxidative damage. It doesn't happen here, and it doesn't even happen, which you might have expected, on the double bond. It happens simply at the bis-allylyl position. So there you are, a very precise sensitivity of the way in which the individual parts of the molecule, not just the molecule, but individual sections of the molecule, are actually of pivotal importance biologically. Now, if we go on, that shows a paper published on the oxidation of linoleic acid, again showing exactly the same position of which the first abstraction of hydrogen takes place to form the peroxidative products. So, if we think about this, that the oxidative products, the superoxides, etc., what they do when they are created, find themselves surrounded by a forest of lipids. And a lot of these lipids are polyunsaturated fatty acids. And if it damages one of them, you then get a chain reaction which will progress throughout the membrane. And a lot of the products of that are pretty toxic, and they will diffuse out and cause havoc in the rest of the cell. However, a friend of mine, Misha, has discovered that if you actually deuterate the bis allylic position, if you put deuterium in it, it hinders the process of peroxidation. And in fact, it'll stop it dead in its tracks. Now, this is unpublished stuff, so I've got to be careful about what we say about it, but it is so interesting because, in fact, where's the next slide? There we are. He has actually shown that they have done this with chemistry. They have a bunch of good chemists have been able to put deuterium in these very specific positions, and it is only this one position that it will be effective. You put it in other positions, in the allyl or the aliphatic positions, and it doesn't have any impact. But you stick it in those allylic positions, and it completely stops the peroxidative progression, which is very interesting. Again, confirming the significance of the very highly specific sections of the molecules, not just the molecule, but individual sections of it, as important in biology. So what is special about docosahexaenoic acid to explain this 600 million year track record in neural signaling? I'll just try and say a few things about it, but you all know about the chemistry of docosahexaenoic acid. You have two docosapentanoic acids, two of them with only differed by two hydrogens, the same chain length, 5 double bonds, just two hydrogens different. And throughout that 500 million year period, although the precursor with docosapentanoic acid in the omega-3 series would be easier to make, thermodynamically simpler, less susceptible to peroxidation, but it was never used in the photoreceptor or the signaling systems of the synapse of the brain. So there's something very special about docosahexaenoic acid. This is getting around to the real meat of the thing. You know, it's just 600 million years and just two hydrogens different. You'd have thought nature would have come up with, or DNA would have come up with some solution to make it easier to make photoreceptors and synapses, but not a bit of it hasn't been working. Only docosahexaenoic acid has been used. And so we've been looking at the molecular dynamics of what's going on in docosahexaenoic acid, and one of the most interesting things is that it has this double bond sequence of which the 1, 3, and 6 double bonds are planar. That means they're in the same plane. And the second thing about that is that it provides a characteristic which, if separated by methylene groups, means that it cannot act in any kind of electrical way. So what is happening here? Why is this so special? If we look at the docosapentanoic acid, there is a significant difference which gives you a hint that the distance between the double bond sequence and the carboxyl is 8.4 angstroms, whereas the distance at the other end is only 4.2 angstroms. And if you look at Hopfield's study of electron tunneling in proteins, which is a means whereby electrons are moved around proteins, it won't happen at 8.2 angstroms, but it will happen at 4.2. So let's just think about what the possibilities might be. Well, that's just the planarness of the molecule. If you look at the electron density in 2 dimensions, using molecular dynamics again, what you find, which is most interesting, is that it's not that the electron cloud is actually active not only around the double bonds, but also around the methyl groups. And this is seen again in the 2-dimensional business, where you actually have an alternative of positive and negative throughout the molecule in a wonderful arrangement, which immediately smacks of some kind of electrical behavior of the molecule that could be one of the forces acting to enable it to operate in a signaling position. So let's look a little bit more about this. Let's think about it. The methylene group interrupts the pi electrons that are present in the double bonds. If you've got a molecule where you have a whole sequence of conjugated double bonds, as you do in retinal, for example, then the electron clouds overlap. And so it actually works like a copper wire. You can send electricity through the molecule without any hindrance. But the interesting thing about docosahexaenoic acid is that you've got these methylene groups in between the double bonds. This means that these electrons are, in effect, isolated in their wells in the double bond sequence. And in classical physics, no current can pass under those sort of conditions. There can be no communication. There can be no cohesion. But in quantum mechanics, all is possible. You can't quantum mechanics is really rather good because it's all full of probability as opposed to the precision which we have in the world that we understand. So really, in effect, what you've got is that these CH2 groups would act as barriers under conventional physics. Now, the sort of barrier you think of is a brick wall. And you've got to have the energy to jump over the barrier to get to the other side. But in quantum mechanics, there is no such thing as a brick wall. All you've got is a kind of an electrical probability. Depending on the electromagnetic properties of the region, the electron will be in one place or another, and the velocity of it can be determined, but you cannot determine where it is. So in effect, it can be a wave or it can be a particle. It has a dual characteristics. And if you cannot determine where it is, the possibility is that it can burrow through these barriers, so-called barriers. And that is feasible. And the beauty about this is that you can actually extract an electron from a double bond within a molecule like docosahexaenoic acid. And if you do that, then the other electrons in the other double bonds can tunnel through to replace that electron if you have a positive and negative dipole moment across the molecule, as there is in the cell membranes. Now, the beautiful thing about this is that, according to the Pauli exclusion principle, no two electrons can have the same properties. They can't have the same energy. There has to be something different about them. There has to be a different spin at the least. You can have two electrons with the same energy, but one has to have different spin to the other. So if you pull an electron out of the molecule, it can only be replaced by an electron from an adjacent system that has the same spin and the same quantum mechanical properties. What this means is that you can only make this electron tunneling business through the energy barriers operate at a single energy level. Now, when you stop and think about this, one of the things that's unexplained about visual reception, and we're not talking about synaptic function for the moment, but one of the things that's unexplained about about photoreception is the absolute precision with which it takes place. If you don't have absolute precision, then the signals are going to be blurred. If it wobbles around an energy level, a range of energy levels, then you're going to get a blurred signal. What does that mean in terms of function? That means loss of visual acuity. That means poor visual acuity. What is one of the first things that you see with docosahexaenoic acid deficiency in babies fed on milks that don't have docosahexaenoic acid is loss of visual acuity. And so this is now beginning to get very interesting because we don't have any explanation as to the absolute precision of the depolarization that takes place, but docosahexaenoic acid, because of this quantum mechanical property, could provide that explanation for absolute precision. Right. Now, there are some things that are currently unexplained without this. The first is, how can you see across a range that is 1 to 10,000-fold different. You can look at a light bulb, a 40-watt light bulb, and read the inscription on it. You can look across the valley at 2 miles away at the same electric light bulb at a distance. Only a pinprick, but you can still see it. A huge range of visual capability. And that, again, can only be explained by the fact that there's a control, an absolute control of the way in which the signal is transmitted from outside to the nervous system. Now, again, the single electron, or single photoreceptor system, where people have fired photons at different energy levels at them, they always get the same response out. That has to be a quantum mechanical thing. There's no quantum decision-making system in the current explanation for photoreception of a whole bunch of GMP proteins and cyclic AMPs and things like that happening. So, now the other thing is that we're going to come to is, why is the photoreceptor upside down? Why doesn't it face the light? Very strange. Well, if you look at the moments of inertia across the molecule, what you find actually is that these methylene-interrupted double bonds, the methylene groups themselves, actually reduce the moments of inertia across the molecule and bring the molecule down to the level at which it's capable of photoreception. It's capable of being operating in photoreception itself. And after all, we have Einstein won his Nobel Prize for his work on the photoelectric effect. So an electron is energized by a photon. It gets into an outer orbit, and if it gets into so much energy that it flies off, then you can get an electrical current moving. And this is exactly what happens with, for example, retinol, where the 11th cis electron is shoved out into an outer orbit, is lost. You get a single bond that twists, then it pulls back the electron and falls back into the all-trans position. So you now have an example of the Einstein photoelectric effect, pivotal to the signaling system in the visual capacity, and what we see here is that docosahexaenoic acid is capable of doing the same thing. And taking an electron out by photoinduction could then lead, with the hyperpolarization, to the effect of what I'm talking about, of electron tunneling, leading to a semiconductor effect and the signal. So sorry, can you go back to this one? Here you have a visualization that if you think of the electron as a ball, it can't go through the tunnel, but it can't go through the barrier, but as a wave, it can. And it'll only do it at the one energy. And we think this is the way that it's working. And we've subjected docosahexaenoic acid to the nuclear Overheiser enhancement, whereby we use a magnet, big magnet in the NMR machine and switch the polarity of the magnet. If there's no electrical activity, no electromagnetic activity in the molecule, then you'll get a straight line. But if there is electromagnetic activity, you get a residual subtracted signal. And that happens with all the positions that we've looked at in docosahexaenoic acid, showing that it can certainly behave in an electrical way. And if we like docosahexaenoic acid, our conclusion here is that it's actually acting as a semiconductor, controlling the signal in the photoreceptor and probably doing something very similar in the synapse. I just want to end by saying that the marine food web, the past and the future of Homo sapiens, well, we evolved from Africa eating seafood and populated the planet by migrating around the coastlines. But today, what is the concern? If we evolved with the use of docosahexaenoic acid from marine food web, we're now looking today at a serious issue. Here's Joe Hibbon's data showing that if mothers eat seafood during pregnancy, they have a greater likelihood of producing more intelligent children. And these are data from 14,000 pregnancies, a stepwise progression of what the mother ate during pregnancy. Looked at the child at 8 years of age. And what we're now seeing at Hammersmith looking at the brain magnetic resonance scans of children, of babies born, we're looking at some quite serious, disturbing findings just by looking at allegedly normal children. We're seeing cysts, we're seeing punctate lesions, we're seeing white matter abnormalities, and we're seeing gray matter abnormalities. And these are in children that are allegedly supposed to be normal. And we're beginning to get very worried about this, and we've got very good reason for being worried, because in fact, the marine wild catch has plateaued, and so eventually the aquaculture must plateau because it depends on the wild catch. The Japanese now have tumbled to this idea and now developed what I call marine agriculture. And agriculture, well, you know about that. They're doing the same thing in the sea. They have pastures for the fish in the sea. And this is the only way we're going to go forward. And we know so little about the marine environment. These are some of the deep sea stuff, which are quite productive. You're pushing worms that are 7 feet long. There has to be a food chain down there despite the lack of sunlight. And that's farming, seabed kelp farming in Indonesia. So it's all possible to produce enough DHA. But at the moment, what's happening is that we're looking at the conditions of existence across the planet in which brain disorders are now risen to overtake all other burdens of ill health. In the European Union in 2004 it was 386 billion, 2010, 789 billion. And the present data for the UK is 105 billion pounds, a greater cost than cancer and heart disease combined. And really the stagnation of fisheries, the source of omega-3 fatty acids, threatens sustainability of humanity and mental health and intelligence. And just remind you about the story is that the brain evolved 500 million years ago in the sea, and it still depends on the omega-3 fatty acids that are in the marine food chain. And we lose sight of this at our peril. Thank you very much indeed.
Thank you, Michael. Michael was one of the most demanded speakers at the last Go It Exchange, and I think that every time I hear him speak, there's amazing amounts of new information that I don't understand, and so I learn something new every time. But I have a few questions here, but I want to be sure we take questions from the rest of the audience as well. But why don't I start with one of my questions? Do you think there's a role, or have you seen any evidence for how other omega-3s, like EPA or DPA, can impact DNA function?
I think the evidence for the other omega-3 fatty acids is for other purposes. I think icosapentaenoic acid, for example, is almost certainly important as far as the immune system is concerned, and particularly as far as the blood vessels are concerned and blood flow. I think what we're looking at here is a multi-generational thing. And a lot of the studies that you're seeing in terms of the role of EPA and the influence, for example, the arachidonic acid eicosanoids, are sort of surgical time points within the tiny spot of somebody's life, whereas what I've been talking about is something like 600 million years of evolution, where it's quite clear that the advantage of having docosahexaenoic acid over all the other fatty acids is very powerful.
Well, actually, my follow-up question actually leads into that then. So have you seen any evidence that the insufficient intakes of omega-3s are leading to permanent structural damage to human DNA? I
think it's, if you look at the comparative data that Andrew and I got on these 42 species, what you see is that the chemistry of the brain is always the same. What's different is the extent to which the brain has evolved. And that fits with the idea that DHA is actually triggering a lot of gene expression for the development of the brain. So in terms of damage to the brain, I don't see it in that framework. I see it in the developmental framework of being actually critical in determining the way in which the brain develops from the human embryo. It's obviously going to be important in terms of maintenance, but I don't see it in terms of damage.
No? Okay. With that, then, thank you, Michael. Thank you very much.